let's get started. Yeah. Again, welcome everyone to this morning or afternoon's webinar, wherever you are. We'll be presenting on Crops to End Hunger project activities or investments in RTB breeding. So, as you know, this initiative is funded by different donors. I've listed them there, mainly the Gates Foundation, Australian government, German Economic Cooperation and Development, GIZ, USAID, and FCDO. So we know the importance of RTB. It's a food security crop for millions of people, particularly in Africa, but also across the world. It provides nutritional security, and many of these crops are actually very resilient to drought and heat. So they're the crops that would survive where other crops would fail in case of severe drought. So farmers or communities that depend on them would always have something that they can rely on. And it's very important in poverty alleviation. But because of the clonal propagation nature, they have some challenges. That's why, as a community, we work together as RTB because we have to share expertise and resources to address some of these common challenges. One of them is the clonal propagation that results in low multiplication rate, lengthy breeding cycle, and some difficulties in scaling of improved varieties because of this low multiplication rate issue. Also, because it's clonally propagated, there's the challenge of restriction in terms of germplasm exchange between regions, even within regions, because of phytosanitary issues. Many of these crops are bulky and perishable. So, for example, a crop like cassava, it has to be processed within 24 hours, sometimes 48 hours after harvest. If you don't do that, then they deteriorate very quickly. We also have other problems or challenges related, for example, to crossing. This is maybe low number of pollination events per cross or low number of seeds per cross. So the investment actually is trying to address many of these issues. And lastly, more important also is the quality traits, because RTB are usually processed into a number of different products, and these products determine the market segment. And most times, this quality evaluations is pushed to the last stages of selection because of the lengthy process that is required to generate those products. So we have some investments that is geared towards addressing this. So for today's webinar, we have 4 people that will present: myself, Xiaofei from CIAT Biodiversity Alliance. We have Morag Ferguson, who will also present, and then Hanele from CIP will present. So we will have, I think, the current time that I'm taking is part of the introduction, 10 minutes. Then Xiaofei will talk about the upgrade of infrastructure and facilities for cassava. Morag will talk about regional germplasm hub that covers all the RTB crops. And then Hanele will talk about investments that is related to potato, the sweet potato breeding network improvements. And then we'll have 20 minutes for question and answer. So without further ado, I would like to invite Xiaofei to start the presentation. Xiaofei, I can roll the slides if you ask me to. Okay?
Yes, please. Thank you. Thank you, Ismail. Yeah. Okay. Yeah. Thank you. Thank you again, Ismail. So yeah, here I will mainly talk about the project on cassava. So it's about we are focusing on accelerating breeding and genetics. So this is a project co-developed with IITA with IITA team. So here I will be mainly present the activities and also progress at CET. So we have four objectives. So in these four objectives, we mainly are going to scale up the tools we developed in the NextGen project, in RDB food project, and also other CGIAR projects. So that they are flower-inducing technology and genomic assistant breeding, rapid propagation, and also the queen quality measurement. So on paper, so these four objectives align well with the two components in the genetic gain equation or the breeder's equation. So that's about the duration of selection cycle and also the accuracy. But also these activities also affect the other components, like after we implement flower inducing, we can make use of the useful diversity from the erect plant type progenitors. So that will also increase the useful genetic diversity for the breeding population, and also for the after we implement the high throughput or medium throughput protocol for queen quality traits measurement, we're also going to increase our selection intensity for queen quality traits. So next slide, please. So I will first talk about the flower inducing technology. So in Cassava, the breeding program have this dilemma. So farmers really like the erect plant type varieties. So this variety usually have very late or don't have branches or flowers. So this give breeders difficulties. That means the breeding program not able to use this good variety as parents to make most use of their useful genetic diversity. So in the NextGen project, we develop a flower inducing technology. So the next slide, we team up with Cornell University, IITA, and also national programs to develop this technology, and we showed the effectiveness of this flower inducing technology. So next slide. So there are four major components or pillars in this flower inducing technology. So the first is about the location. So usually it's the location of the crossing nursery. So would it be ideal to put the crossing nursery in the medium altitude location about at least several hundred meters above the sea level. So the second component is photoperiod extension. So we implement the red light to extend the photoperiod to have earlier flowering. So the third component is about pruning. So we prune the young stems in the very early stage. So when the young branches, when the branches is even less than 1 centimeters, we cut them off, and to let the the flowers grow. So after pruning, we applied the hormone to increase the fertility of the flowers and also convert the flower from male flowers to female flowers. So that we know only female flowers can give us fruits and produce more seeds. So next slide, please. So here just one slide to show you the effect of the photoperiod extension. So on the x-axis, you can see that's the days of the first branch, or we can consider it's the days to reach flowering. So and on the top you can see a lot of progenitors under natural light. That's the blue dot. So it requires 7 or 8 months to have the first branch of flowering. But under red light or photoperiod extension, that's the orange dot. So they only require 3 or 4 months to get flowers. So this is dramatic. This will dramatically help the breeding program to have early flowers, to shorten the pollination season and potentially shorten the cycle of the breeding program. So the next slide, please. So in this project, the objective one, we mainly focus on flower inducing again. So we are going to mainstream this technology, and we are going to establish a 0.5 hectare, a 0.5 hectare red light area for cross nursery. And in next year, we are going to schedule a training workshop with our Africa partners to scale up this flower inducing technology with national programs. So after we generate the seeds, we usually every year we have some seeds left over. So would it be good to put the seed in the good storage condition? So this is another activity we are doing, is we renovate our seed storage room, so we can control the humidity very well. So and then we can maintain the long period of storage and also maintain the activity of the seeds to share with partners. So in the slides, I use green color to show the activity we already finished, and the blank color is the activity we are still going. So here is just show you the 0.5 hectare red light area we used for cross nursery. And on the right, you can see after implementing the flower inducing technology, we have much larger inflorescence, and we have more female flowers and more fruits. The second objective is mainly about genomic assistant breeding. Besides the genome selection at CIP, we also work on an initiative is about using inbred progenitor-based hybrid cassava breeding. So the first, we are studying inbreeding depression. So now with the reduced the sequencing cost, so we are able to afford using whole genome sequencing to help us to answer some questions about large effect dilution mutations, about inbreeding depressions in cassava, and also this whole genome sequencing will help us to discover traits to identify the QTL, or even narrow down to several candidate genes on several traits like dry matter content, like plant architecture, like the cassava brown streak disease resistance. So we team up with BGI. We have collaborative agreement. Then they provide a service for us to do 10-time coverage whole genome sequencing. Next slide, please, Ismail. So we sequenced our parents, the germ section population, and also the genetic study population for inbreeding depression for trait discovery. So every year we have about 3,000 samples from this 10-time whole genome sequencing. So that's about 24 TB data. So we need to increase our capacity to manage this data, to analyze this sequence data. So I think now in the 1CG we are still we are still discussing how we are going to using the shared server, the shared computing system. So I think at least in the next 3 or 5 years, this server is still very critical for us to start to implement this to implement the genomic assistant breeding and to make use of the genome sequence data. So the next slide, and we are also send our team members to different workshops. So one is in University of Florida, the other is the Summer Institute, organized by the Plant Breeding Center in University of Minnesota. So we have team member there to join the workshop to really increase their professional skills in quantitative genetics and also in breeding. We also invite experts like Diego and Gay to come to CIP to give a workshop about germ selection, about quantitative genetics. So this is kind of all these activities kind of co-funded with other project at CIP. So the next slide. The third component is about queen quality. Is also aligned well with what Ismail mentioned. Queen quality is very important for the increase the adoption of the new varieties. So here we are purchase equipment and also innovate our facilities to scale up several tools we developed in the RTP food project. The first one is hyperspectral imaging. So in RTP food project, we already we confirmed that this technology can not only measure the dry matter content, but it also shows the distribution of the dry matter. So you can see the red color means high dry matter, and the green color is show the lower dry matter. So this tool can help the breeding program to select for not only high dry matter but also even distributed dry matter content. This will probably contribute to the dry matter stability in the release cassava variety. So the next is about commercial. No, not the next slide, please. So the next is about commercial kitchen. So again, in the RTP food project, we developed a protocol, or the technology is using water absorption to measure cooking time and also mealiness. So now we are building a commercial kitchen that will increase our throughput or our capacity to screen breeding population for water absorption. That will also help us to increase our selection intensity and also accuracy in queen quality evaluation. So, and also we plan to schedule a workshop in 2024 to scale up these queen quality measurement protocols with Africa partners. So next, I'm going to using several slides to talk about the PPD. So at CIP, we have been studying PPD for many years. We also accumulate a lot of data. So here is one set of the data I showed the repeatability in this population. So after combined 14 environments data, we have the broad sense heritability or repeatability about 0.7. So if we only use 2 or 3 environment data, the heritability will be very low. So that means there is an environment noise there to affect our measurement. So after the next slide, please. After we reviewed our protocol, we realized that there is one step. I used the red the red frame to show that. So after we cut the roots, we store the root for 7 days. So the storage happens in the open area. So there's no control on humidity, no control on temperature or airflow. So I think this varied environment might cause environmental variation and to reduce our accuracy in the PPD evaluation. So in this project, we are going to renovate storage room with controlled humidity, with controlled temperature, and also airflow. Hope to increase our accuracy in this PPD evaluation. So the next slide is about a net house. So it's also aligned well with the propagation, which Ismail mentioned is one of the challenging for cassava. So due to the clonal propagation, is very easy to spread the diseases. So at CIP on campus, we have one disease is called cassava frost skin disease. It's present on campus, but is not widely reported in the cassava production area. So we implemented this net house, try to maintain the material clean, or either the breeding population or the release variety clean. Then we can deliver or distribute the clean stems to national programs or to partners to reduce the spread of the disease or insect. So now we have the 0.4 hectares of net house ready to plant the release variety and also the breeding material there. So I will kind of summarize what I just presented. So we have 4 objectives. So the first one is about the flower inducing technology. The second one is about the server to increase our capacity in genomic assistant breeding. The third is about the queen quality, measurement capacity, and also include the PPD measurement. So the fourth is about this net house to increase our capacity for propagation to provide clean material. So I will stop here to give the floor to Ismail to talk about the IT activities. Thank you. Yeah, we can hear you.
Yeah. So most of the previous activities that Xiaofei presented, we are co-implementing. So we have copies of the same activities also here. But now I would just like to spend a few minutes to talk about the special activities that are related to cassava for processing and phenotyping facility upgrade for that purpose. So we have in Nigeria, for example, 4 different major product profiles, and these product profiles are defined by some key attributes. So we have cassava, processed cassava that are processed into different products like gari and fufu. Then we have key traits there, texture, color, and then product conversion rate. That's the quantity of product you get from a unit of fresh fruits. We have the fresh market segment, we have the biofortified segment, and we have cassava for industry that also has a certain attributes peculiar to it. So in order to assess the quality traits for these products, we have to process the root. And for some market segment like fresh, it's very easy because it's just one step, boil or steam. For the starch and so on, there's a multiple steps, but the most complicated ones are processing for example, the main product here in West Africa, that is gari. It takes several steps. And then once we have these products, we have to evaluate them for color, for texture, and then a sensory panel, consumer panel, and so on. But we can only do that at the moment, at the last stages of testing, because of the difficulty and complexity of this process. So one of the investments, really a big one for us, is how to streamline this, have equipment that can make this process much faster. So typically, just one of the product, for example, here is, we get a specific quantity of roots, peel them, grate them, dewater them, and then toast in dry fire, and then you get the granulated product. So we want to move from the previous way of doing things, which is more like a traditional processing facility, to a more streamlined factory-like workflow, where we have equipments installed and everything is laid out in a sequence that flows very easily, so that we can increase our throughput. And these are just some of the example of equipment for peeling, for example, instead of hand peeling, using machines, grating, sieving, and also even the toasting, to use a mechanical toaster, and then a good facility for storage. So we have been working with our architects to design the workflow. So everything looks a bit streamlined now, and the construction is going on, and hopefully we will be able to commission the facility early next year. And this is just a rendering of how it will look like. So what do we expect after this implementation? We expect that we can deliver varieties that are suitable for our product profiles and market segments much faster by increasing this operational excellence, evaluating more lines early during the early stages of testing, standardize the processing, and also it's good for the people that are working in the facility because it ensures operational safety and then hygiene of the product and so on. So we're very grateful for this particular investment. So now Morag will give update on the Germplasm hub for vegetatively propagated crops. So Morag, I don't know, do you want me to unshare and then you load from your side, or I can just continue rolling the slides for you?
I think let me try and share myself, huh? If you unshare. Okay. Okay, can you see my screen?
Mm-hmm. Put it on present on slideshow.
Yeah, we can, but it's not on.
I'm putting it there. Is that good?
Yeah, this is okay. Thank you very much.
Okay. Okay, thank you very much. So yes, thank you very much, Ismail. So I'm going to be talking about an investment which is with KEPHIS, which is the Kenya Plant Health Inspectorate Service, and the CGIAR. It's a partnership for a germplasm hub for roots, tubers, and bananas. So KEPHIS, as I said, the Kenya Plant Health Inspectorate Service, this is a government parastatal. It's the national PPO, the Plant Protection Organization, and its role is to really assure the quality of agricultural inputs and produce, to prevent adverse impact on the economy and the environment and human health in Kenya. So this is also a partnership between CIP and IITA and KEPHIS, and actually CIP and IITA have been working with KEPHIS for the last 20 years. So it's really not a new partnership. And we'll be talking about sweet potato, cassava, yams, and bananas. So I know Ismail talked a little bit about the importance of root tubers and bananas. And again, just to reiterate, they're very important in terms of food security, high yielders in terms of calories. More than 300 million people below the poverty line depend on RTBs for food and income. They're climate resilient, nutrition security, often rich in key nutrients such as provitamin A. They're frequently grown and marketed by women and can also be processed into many food products and have a lot of industrial applications for income generation. This is in terms of high quality flours for porridge or blending or for bread making or confectionaries, weaning foods. Cassava is a very important source of starch. Often the peels and the byproducts can be used as animal feed. They can be used for biodegradable packaging, which is very important as we move away from plastics, as well as ethanol production. So apart from the food security, there's tremendous potential for income generation. So I just wanted to share with you a little bit about the importance of and the contribution of RTBs. So here we have 6 countries: DRC, Ivory Coast, Ghana, Nigeria, Rwanda, and Uganda, and their populations. This is 2017 data, and the total number of calories consumed. Then we have those calories consumed from RTB foods and the percentage of foods of calories from RTB foods. And you'll see that in these 6 countries, 40.5% of calories are obtained from RTB crops. And with this population, it's predicted to almost double in the next 25 years. So if 40% of calories are coming from RTBs, we really need to improve the productivity of RTBs in these countries and many of the African countries. So one way of doing this is really to get improved varieties out to farmers much quicker than we're doing at the moment. We have to recognize that not all breeding programs in all countries are well-resourced. And so we really need to try and leverage the stronger breeding programs to help the less resourced breeding programs. So one way of actually getting the material out to these a broader range of countries is really to get them to maybe test mid-stage breeding material, not necessarily wait for finished varieties, but test perhaps at the UYT stage. This will really enable NARS to select under their local conditions, with local preferences and local market segments. It will also mean that the breeder's seed will be bulked early, and varieties can then be entered into NPTs early and reach farmers much more quickly than they're currently doing. Now, one issue, as Ismail has already touched on, is there's a challenge to germplasm movement with clonally propagated crops. Clonal propagation means that they don't normally go through a conventional seed stage. And so a lot of the pathogens are carried over from one season to the next. Virus elimination and pathogen elimination can be very time consuming. And this, even pathogen infection or the potential for infection, really delays the regional distribution of planting material. This is also compounded by low multiplication rates. So IITA and CIP, as I said, we've been working together for the last 20 years, doing virus elimination. We've been working with KEPHIS, doing virus elimination and moving germplasm around the region. KEPHIS is a regional quarantine station, but the facilities we've been using were actually built in about the 1970s and have really are really not fit for purpose anymore. And we've been getting quite a lot of problems with contamination, and the facilities are really too small as well. So it was agreed that we should apply for funding from the Crops to End Hunger for a centralized facility within KEPHIS that can operate at scale and provide RTB germplasm services to the CG-NARS crop networks and really serve as a center of excellence and training. And this really aligns very well with the vision of a stronger CGIL-NARS breeding networks, where some of the downstream functions are operated by NARS and the regional capacity is developed to maintain and distribute elite germplasm. The CGI will obviously continue to backstop in a number of areas, but will gradually transition to be more of a service user than a provider. So some of the services that are going to be offered by this new facility, first of all, receiving germplasm from breeding programs across the RTB crops. There'll be internationally accredited diagnostics, pathogen elimination, in vitro tissue culture facilities, tracking systems to ensure that, when a request is made for germplasm, that the correct germplasm is actually distributed to maintain integrity of the germplasm. There'll be genotyping for quality control. And as KEPHIS is already a regional distributor and quarantine station, they actually provide the import permits and the phytosanitary certificates. So that is routine for them. So that should make the flow much easier. There will also be some capacity building to ensure that the network nodes are capable of receiving the germplasm and hardening the germplasm to establish it well. So we started with a high-level project initiation meeting with KEPHIS management on the 3rd of March, in which Ian Barker, Senior Director of Strategy, Delivery and Scaling, visited senior management at KEPHIS headquarters. We then had a project implementation initiation meeting on the 20th of June with the team from KEPHIS, CIP, and IITA. And we've divided the implementation into 8 different modules. These are firstly building design and planning, building implementation, equipment, systems development, operational business plan, partnerships, capacity building, and project administration. So this is a fairly sort of complex project, and I'll just go through some of these different modules. So one of the first things we had to do was obviously site selection. So this is the site at Muguga, which is the plant quarantine and biosecurity station of KEPHIS. And you can see in the blue, that's where the site is. And you can see some existing buildings, which are quite traditional buildings. We also had a topographical survey done, and we then scoped 4 different architectural service providers and came up with, we unanimously chose DMJ Architects. This was also done in collaboration with the Breeding Resources Initiative. We then got started working on the design of the building and had quite a lot of consultation with KEPHIS, CIP, and IITA, and particularly discussed the needs of the women employees, and also got initial support through the architects in Brazil through Breeding Resources Initiative. The design was very much based on workflows of potatoes, sweet potato, cassava, banana, and yams. So here we have a schematic of the building that is planned. As you can see, it's a sort of modern take on a traditional design, and we have a walkway around the building so that visitors can see what's going on through the windows, but we keep the inside as clean and sterile as we possibly can. So we've pretty much got to the final adjustments and the design of the building, and we've engaged with service and structural engineers and are planning to break ground in January. The idea is that we will complete by December 2024, but this is going to be challenging. So one of the major things is we really want to make this building as sustainable as possible. And at the moment, KEPHIS is actually paying about $5,000 per month for electricity. And this is really taking away resources from other areas of work that should be supported. So in this building, we're aiming for about 90% of electrical demand to come from solar power. We have engaged a company called Rome to provide a lock and key solution to this, and they've installed a power logger to determine the current energy consumption and the patterns of energy. And it's projected that the energy needs are about 200 kilowatt hours at peak consumption, which will need about 348 solar panels, which will take about 1/4 of an acre. So we've decided, we've gone through lots of debates about whether we should be utilizing the roof space or the solar farm, but apparently the angle of the solar panels is very important. So we're tending towards a solar farm at the moment, and a space has been allocated for this. Now, there's also a number of outbuildings that we need to, some of them we need to renovate. So there is a caretaker's house, which is very close to the site of the new building. So we're going to have to relocate the caretaker, and we're going to have to renovate another building on site for him and his family. We would like to renovate the existing caretaker's house for administration and finance officers. And then there's also a canteen area, which we would like to renovate and refurbish, and actually add a room for a women's restroom for pregnant women or breastfeeding women. We'll just have to see how the budget goes as we move a little bit forward into the project. Another area that we've had to deal with is aligning with the government. As you know, KEPHIS is a government parastatal, so we have to align with the Ministry of Agriculture and Livestock Development, and also engage the Ministry of Public Works. So to do this, we had a meeting from the 24th to the 25th of August, just outside Nairobi, which was kindly funded by KEPHIS. So we are now fully aligned with the government, which is important. We've got a list of equipment that we continue to refine, and the major equipment has been located on the floor plan of the building and listed by room. And CIP has initiated the ordering process in September 2023. We're also working on systems operations, and this component is being coordinated by Trusha Shah, implemented by Margaret Karanja, who's based in Nairobi at IITA. And KEPHIS have nominated 3 employees to work with them on the implementation of this. And this basically involves mapping the workflows and data flows, standardization of vocabulary, ontologies, SOPs, etc., digitalization, batch processing, which includes a lot of capacity building as well, and IT infrastructure, such as database schemas, looking at printers and labels, etc., etc. A fifth area, is the operational business plan. So clearly we want to make this lab as sustainable as possible. So the activities and the processes need to be costed out accurately, and a sustainable business plan developed so that we can have chargebacks for various services offered. So this component is being coordinated by Srini from CIP, and KEPHIS again have nominated 3 people to work with Srini on this aspect. So, you know, the partnership, I think, with KEPHIS is very important. And we need to define how this partnership is going to work. We signed an initial broad MOU in September this year, which gives a broad outline of our intent to collaborate. But we're working on a more specific project focused MOU to see how we're really going to interact with this. Clearly, the lab will be jointly managed by KEPHIS and CGIAR, which will be reflected in the name, I think. And then also CG will provide backstopping in terms of protocols, purchasing, networking with potential clients, the NARS breeding networks, in providing germplasm and supporting IT systems. So again, capacity building is an important component. And already 2 participants from KEPHIS have been over to CIP in Lima, Peru, in September to conduct training in various aspects of the lab operations, including in vitro clonal crop production, diagnostics, phytosanitation, genotyping, data analysis, distribution, and quality control. We have also started interacting with the NARS breeding networks and given a number of presentations about the lab. So that's all from me at the moment. So thank you very much for listening, and I look forward to hearing any suggestions, comments, or questions. Thank you.
Thank you very much, Morag. We have time, but I would prefer that we finish the presentations and then we can go to the discussions at the end. We're doing very good in terms of time. So maybe you can stop sharing and then Hanele can share her slides. Thank you.
Thank you, Ismail. Let me try to share my screen. Am I sharing the correct screen now?
Yes.
Yes, okay, great, thank you. So, for those who don't know me yet, my name is Hanele Lindqvist-Kröse. I am leading the sweet potato and potato breeding team at CIP. And I'm presenting here on behalf of the team. So my colleagues Bert de Boek, Elisa Salas, Tiago Mendes, and Jan Kröse have been very much involved in all of these activities and planning. So we at CIP, we are involved in 4 of the projects of the Crops to End Hunger. So the first one, the project number 14, is investing in sweet potato breeding networks to mitigate climate change. And here we are, the facilities that we are improving are mostly or are in our own breeding station in Lima and in Huancayo. Then the second project is upgrading the CIP-NARS East Africa potato breeding hub infrastructure, where this is in Nairobi, where we work at the facilities of partners. So we are investing in the facilities at Calro and Tigoni and University of Nairobi, at the Kabete campus, and also we are using some of the facilities at KEPHIS, like greenhouse facilities. Then we are also part of the project, like Morag presented, the regional germplasm hub for vegetatively propagated crops. And like Morag very nicely presented, this is focusing on KEPHIS in Nairobi. Bert is also involved in this project about global breeding analytics unit to accelerate the delivery of superior genetic gains on farmers' fields. This is in collaboration with all the CG centers. And in the slides that I have put together, I'm focusing on the first 2 projects. So the goals of this project are to focusing on the different components of the breeder's equation. So we want to shorten the cycle length, and for that we are improving the true seed processing laboratory and also the greenhouse spaces. We are working on the better facilities for tuber seed multiplication and storage. To increase the selection accuracy, we are investing in digital tools for data collection with NARS partners, as well as for our own breeding program. Digital scales, and we are improving the processing laboratory that is focusing on the quality traits evaluation. And to improve the selection intensity, we are introducing some high throughput platform for selecting traits. So we're purchasing the smart grader for yield assessment in potato. And we are investing in new greenhouse facilities for crossing blocks. So all of the kind of cross-cutting activities here are capacity sharing, capacity development when we introduce something new, the digital tools, for example. This comes together with training events. Also all the different equipment and so on that we are introducing requires capacity development. We also want to focus on gender equality and climate change mitigation. And an example of the gender equality that would be that the person's physical strength or height would not be a limitation so that they can access a tuber seed crates, for example. And for climate change mitigation, we are looking at things like improved water use efficiency and how we use fertilizers. We are recycling the soil substrate in the greenhouses and things like that. So the objective is to improve selection accuracy, shorter breeding cycle, and better selection intensity. So we want to do this by increasing the capacity of generating new crosses and extract true seed, increase the capacity for multiplication and storage of tuber seed, and more precise and faster genotyping of priority target product profile traits. And so we have some goals. When we evaluate larger population sizes, we can use higher selection intensity. And this was actually a BPAT recommendation a couple of years back. When we can evaluate the key traits in earlier stages, we want to, our long-term goal is to shorten the breeding cycle by 4 years. It's not only going to be done with these infrastructure improvements, but it's part of the breeding scheme optimization, use of molecular markers, genomic selection. And we want to increase the heritability of some traits. And here is an example of the dry matter content heritability should be, the goal is to increase it by 10%. So some of the challenges in potato breeding, so the improvements or these projects are mostly focusing on potato. And so that's why I'm talking about potato here. So the challenge in crossing, we have some challenges in making the crosses in potato. If we look at the pictures here and this scheme, it seems pretty straightforward and not that complex at all. But if we go into the more of the details, how to manage the crossing block, we notice that there is a lot of manual labor involved and it takes a lot of time. So whenever we can improve the workflows, it liberates the people from these kind of activities to do something else, and we can be much more efficient. So the whole crossing block takes around 8 months. we start by planting the progenitors, first planting the male plants, and 1 month later the females. So we prepare the greenhouse for the crosses. We set up the bamboo stakes that will be support for the plants, and pots with the soil. Then after, when the plants have nicely emerged, they have a nice root system, we set them up on top of these pots. We don't bury them into the soil, because there needs to be the next activity is we have to manage the plants after they grow. So the stolons must be cut so that the plant directs all the energy towards flowering. And as they grow, we need to tie them for support in these bamboo stakes. So potato has a lot of different diseases, and it's important that we have these crossing blocks inside in greenhouse so that the plants stay healthy. And also we do disease detection at this stage. So we do serological tests and PCR tests to make sure that we don't have any seed transmitted diseases there. So there are 2 viruses that are seed transmitted, and if we detect them, then everything has to be destroyed, and for sure the true seed from these crosses cannot be distributed anywhere. So then as the plants grow, we start collecting the pollen. We have to analyze the viability, and then we make the crosses. So the flowering period lasts between 2 and 3 months, and everything is done manually. So first there is the emasculation, removing the anthers, and processing the pollen, drying, and so on, and then do the fertilization. Then after this, we collect the berries and process the botanical seed. So the good thing about potato is that it produces a lot of seed. So at least that is something positive. But it takes around 2 months to process the botanical seed. So we want to shorten the breeding cycle through improved crossing facilities. On the right-hand side in this picture, you can see the current greenhouse structures that we have, and they're quite small. So people are spending a lot of time in moving from one greenhouse to another, which also, again, then increases the risk of diseases coming and spreading, and just takes extra time. So we are building a new greenhouse in our breeding station in the highlands in Huancayo, and also a greenhouse in Calro, Tigoni for the crossing block. So the idea here is that when we have more space with better facilities, we can make more successful process. So I mentioned that it takes about 2 months to process all that true seed. So we want to improve the laboratory. Current facilities that we have are not adequate for enough people to work in them and have a proper workflow. So we expect with this laboratory remodeling to spend 20% less time in seed processing. So the second challenge of vegetatively propagated crops, like my colleagues have already mentioned before, is the vegetative seed production. So this is actually what we plant in the fields for multi-location trials. So potato has a quite low multiplication rate. So one plant produces maximum 10 tubers per plant that can be used as seed. The best quality and disease-free seed needs to be produced in seed beds, again in controlled conditions and in the highlands, because potato has lots of diseases, and when there are insects around, there will be viruses. So it has to be in a place where there is less insect pressure. So the current system that we have is insufficient and it's labor intensive because, for example, there is no automatic irrigation. So again, people spend a lot of time watering manually. I mentioned that we recycle the substrate. So of course we need to sterilize the soil, and our current methods are suboptimal for this, again taking a lot of time. And tuber storage facilities are too small. So we want to increase the capacity for tuber seed multiplication. We have already these beds that are on the left-hand side in the picture. We want to increase them, so we're going to put in several more of these beds and install an automated irrigation. And this will decrease the time that people have to spend in manual labor over there. We also, when we add more of the beds, we are going to increase the seed production capacity by 40%. So I mentioned that we are recycling the substrate that we use in those beds. So one of the bottlenecks that we have is the soil sterilization. So we want to install this kind of a steam producing system that sterilizes the soil. And we actually want to have this sustainable. It should work with solar energy. But as the company that was making the plans for this came to look at our facilities, they said that the total renovation of the electric circuits at the same station needs to be improved so that this machine can run with electricity and that we can switch to solar energy. So at this moment, I'm not sure the project has enough funds to do this. So the seed storage facilities are being improved. So we are building these diffuse light storage facilities both in our campus in Peru and also at Cairo and University of Nairobi. And we are expecting to increment the seed storage capacity by 25%. Another thing to approve is the selection accuracy. So we want to be more precise and faster in our genotyping. And that's why we are purchasing more digital tools for phenotypic data recording in the field. And then we are sharing this with the NARS partners in Bangladesh and also in the East Africa breeding network. We are also buying this kind of digital scales and the smart grader for faster sorting and evaluating yield in data. Part of this is also the quality laboratory remodeling. So I'm not sharing all the details here at this moment. So we have also the objective to strengthen the NARS CIP breeding program's data collection systems and capacity for the digital data collection. And this is important to reduce the errors and improve the quality of the phenotypic data. So we want to have this data collection network with a centralized database that all partners can use. So Potato uses BreedBase. So we have a potato base available, and we want to train at least 40 staff members of the breeding networks for the use of BreedBase and these new digital tools, and also to understand the SOPs that we have already developed for breeding data management. The goal is to have at least 50% of these trainees women. We are also training CIP staff members who work on sensory analysis and organoleptic evaluations and the use of the new equipment in the quality and nutrition lab. And they will then continue training the other staff using the train-to-trainer approach. Morag already mentioned this. So this is part of the KEPHIS Regional Germplasm Hub. So CIP was responsible of training some of the staff in virus diagnostics and phytosanitation. So just mentioning it here as one of the trainings that have already been conducted. So in summary, the project that the Crops to End Hunger project that CIP is involved in is aiming at the increased capacity for generating new process and extract through seed, the increased capacity for multiplication and storage of tuber seed, and more precise and faster phenotyping for priority target product profile traits. And thanks very much for your attention.
Thank you very much, Hanel. So we have quite a bit of time till the end of this webinar. Maybe I can just try to summarize. I think we have had a lot of interesting and exciting initiatives supported by this Crop to End Hunger project. Basically, starting from breeding, looking at how we can accelerate breeding, increase flowering, seed management, phenotyping, and germplasm storage, germplasm exchange. So the whole so-called, so to speak, value chain of breeding is covered. So I'd like to open the floor for questions. There have been some exchanges in the chat, but if you want to ask questions, maybe this would be the time. Feel free just to chip in. I can't see any hand.
Hi, Gary here.
Yes, Gary. Yeah. Go ahead, Gary.
Oh, sorry. There's been some discussion, particularly from Jim Lorenzen, regarding using true seed progenies to reduce for rapid cycle genomic selection, distributing those for tuber increase or vegetative propagule increase at the partner sites, and then using those for training GS models and genomic prediction. Is there any are there any projects on that ongoing? You would need relatively high seed production numbers, which it would work quite well, I think, in potato. I'm not sure about sweet potato or cassava. It would be very difficult, I think, in banana. And any work in that area being done?
Maybe I can start. Yeah, for cassava, we can generate a large number, even though crossing is difficult, but in the right environment, with the right team, with also the flowering, we can generate a lot of seeds. There's no, like the way you have put it, where we generate the seeds and then share the seeds across partners for rapid cycle GS. I wouldn't say that we have done that or any project is ongoing is covering that. But for IIT, for example, when we started NextGen, when we had our seedling nursery, at that stage one of testing, each family we split it into like 3 or 4, and put it in 4 different environments, of course, with the checks connecting them. And that was trying to capture all the environments that we're breeding for. But certainly to address the issue of VPC, or vegetatively propagated crop phytosanitary bottlenecks, I think that would be an option. Maybe if my colleagues, they want to chip in, please, Xiaofei, Anele, Morag, and other RTB breeders that are in the house, please feel free to contribute.
Yeah, so this is Xiaofei from CIP. So we have started some pioneer activity with Asia program. We send the seeds to them, and then they propagate the seeds to put the seed in the field in their environment to collect the data, to send the data back to us to train the model. So I think we have some difficulties in propagate the material in national program. So probably we have, I think that, so this might be a good way to implement genome section with partners, but we need rethinking about the logistics, how we are going to help the partners to get the seed germinated and propagated to get high quality data. So I think so we started, but we have not get some positive data back.
So we might need a specific project to pilot that and help build the capacity in those areas with the key partners who we're going to help. I think
that,
which would probably be a good thing.
Yes.
Yeah, so I can just mention that we started an in-house experiment like that we were sending through seed from a population that we were doing. We had genotype here, and then we send the half SIPs to the breeding program, SIP breeding program in Kenya. And that is still ongoing. So we want to test if the model that was calibrated in Peru for late blight resistance, if it can predict the late blight resistance in Kenya. I don't know if, I don't want to put Mokhtar on the spot, but if he already has some results on that. But yeah, I do think that the true seed dissemination is a very good way to advance work, and we do send quite frequently a true seed to partners. So Mokhtar, if you want to comment.
Yes, Hanele, we had the genotype, also the material that was sent to Kenya. And right now, for example, for the same cycle, we have advanced clones for the let's say the breeding that was done in Peru, and in Kenya, the cycle is still advancing, but we have all the genotypes, all the individuals genotyped, and we are still processing the data with Guy. And last month, he showed very promising results, and we will just catch up next week to see how we can publish some of the information we already have. So very soon we will publish some data regarding this effort. But yeah, the data is very promising, like trying to predict from what we have in Peru, a population that is selected in Kenya.
Thank you, Mokhtar. Maybe I can just add that in our case, it's we use that for preemptive breeding for brown streak disease. So cassava brown streak is mainly restricted to East Africa. We don't have it in West Africa, thank God, so far, but we need to be prepared and to preemptively breed for that. So we make crosses here. We send the progenies to East Africa. They are screened for resistance, and it's both genotypes, so we can see if we can use prediction from one location to select for enrichment of the resistance alleles. So thank you. I
Right.
can see Chedozi.
Yes, thank you, Smart. Thank you, Shofe, Morag, and Hanel. Very great contributions. And thank you, Gary, for all the questions. Very enlightening and provocative. So helping us to focus better. I want to just add my voice to say this is a great initiative, and it's exciting because it's a lot it shows how capacity had degraded over the years, and that it can be done. I saw those pictures by Morag. I said, oh my God. And the new thing that's coming up. So all of these are meant to improve breeding at the local level. I'm just like, how do we extend this? We usually use the word scale. It doesn't have to be very perfect before we begin to implement some of this in the national programs, just like Morag rightly demonstrated, and Hanel also. For the root and tuber crops, this is very important because it's already a crop that is tough for even the CG to breed. And a few national programs who are really doing the beat of developing populations, this is an important thing that we should be thinking about. The crops to end hunger should not end with the CG. It should really get to the NAS where the rubber hits the road. Thank you for everything.
Sorry, I couldn't agree more. So we have the team here, and I think it's very important to make sure that we extend this. Many of these facilities will be shared, of course, but also if there's need, we need to actually put some of these facilities in the local places where things are happening as well. Good. Yeah, please. Any other question or interaction?
So Ismail, chat with you.
Yes. Hello.
Hi, can you hear me?
Yes, we can hear you.
So yes, Chagwe here from Potato Breeder for Seed based in Kenya. So I just want to compliment and also just echo what Gary just said. I think in potato, especially in Africa, that we're expanding our operation here, and we have now a breeding network for East Africa. I think it's just a great opportunity to maybe have a project like you just mentioned that we can extend, be more lean here in Africa, and have the partners running the program, and then we can provide this kind of support and perhaps having the genomic selection as part of the breeding process here. I think because of that, we have this capacity in Kenya now and making the crosses and perhaps working closely with the NARS. The genomic selection would be one of the approach that you can take. But we have to think about what would be the best strategy, sending the seeds out, selecting, making the process again and send it out, because to recover the parents back here, maybe it's going to be very easy to do, especially what Morag just presented. We can receive the parents fast here in Kenya and can make the cross again and recombine the populations, not just for potato, for all the RTB. I think all these projects are complementing well, and we are in a very good moment to try out new technologies, new way to do, new way to connect the networks. Just to make this comment, how we are in a good momentum for that.
Excellent comment, Tiago. Okay.
If I could just add one or two points. If we were going to do that, there's a couple things we need to think about generally about using genomic selection effectively. One would be that if we were going to distribute true seed and then have partners increase clones, test, and train the models, from those phenotypes, then the genotyping would need to be done on those clones, I think, not on the true seed progenies that were sent out. So that you'd have to be sure that you had a system for getting the genotypes the clones. The other observation I wanted to make was, the key tool for increasing accuracy from a pipeline design approach is sampling more environments in stage 1, which genomic selection is very helpful with. So when we're thinking about how to do that, it seems to me that most of these pipelines probably have fairly large stage 2, stage 3 testing networks, right? Testing clones at more locations. The easiest way to increase the size of your stage 1 testing network would be just send some clones to each of your stage 2, stage 3 locations. Is that being done, and is it feasible? We really want to see more emphasis on phenotyping at stage 1 in a broader sample of the TPE, because that's where the great bulk of the selection intensity is applied. And that's where we've got most of the genetic variability still in play to train the model. So those are just two thoughts regarding moving forward with genomic selection. One is if you do move to true seed distribution, you would need to genotype the clones extracted from the true seed, not the true seed progenies. And the other is, the other is a question, are we in a position to be able to distribute these stage 1 populations to most of the locations that are doing later stage testing?
So Gary, that is difficult because of the phytosanitary regulations. So it can be done within a single country, I think, but across borders, that is very difficult, because especially if we try to send anything from Peru to anywhere else, they have to have this phytosanitary status that they are cleared for international distribution. It takes years to get to that stage.
So you're not sending later stage material for late stage trials from Peru. That's all increased in country.
No, we do send for late stage testing. So, well, variety extraction. So those are already advanced clones. Then they are the best that we have selected here. So those would be for variety selection, just because it takes such a long time to clean them up. So the true seed is really the only way to speed things up.
Right.
The true seed progenies are the ones that will be the clones as well. So the genotypes are already fixed from the beginning. We always work with F1.
Yeah, actually, you could use that information from the true seeds, from the locations where we send the seeds, to optimize the cross for a target location, for instance. Maybe what we haven't done, for instance. That's what we could possibly do when you send out the seeds. Optimize the cross for specific target locations. And eventually, as I was just mentioning, in the region, receiving the most advanced clones selected in a given country and clean them up here in Kenya or whatever the place. And then we can design crosses for specific countries. There will be cases when you have like a homogeneous TPE that for traits like late blight, high heritability, we can for sure leverage from genomic model. So it's still a question how we can better connect different countries for yield. But good challenge to think about and see how we can speed up those things using the new technologies. Yeah. But the phenotyping for sure is the biggest challenge for RTB, especially for potato. In the early stage, the plots are very small. When you're thinking about more complex traits, it's very difficult to get high heritability, high heritabilities. So for traits like late blight, small plots is not a problem, but for yield, it's always a big challenge for RTB crops.
We'll come back to this discussion. I think it's important, but Hugo has his hands up. Hugo, go ahead, please.
Yes, Ismail, thank you, and thank to all speakers. I think that it's very exciting to see the progress being made across different crops, and that just building on some comments made by Gidoji and Gary. I think that even though we're not doing that on a regular basis across our crops, but in addition to cassava, potato, we're seriously considering using seeds to be distributed because of the work being on developing hybrids. So, and our plans going forward is to, while where phytosanitary guidelines allow us, is to move as quickly as possible through sharing seeds families because of what has been mentioned. But what I wanted to add it is that in many cases, getting the genotypic information is not the main constraint. The main constraint is getting good quality during the stage 1 at the with the high heritability in the TPE, and but we can achieve that by deploying as quickly as we can spare testing, because that will allow us not only to have a less plots in the field, but also we will be exploiting information for the pedigree, either to the pedigree or to the genomic relationship. So, in other words, going forward, yes, Gary, we want to move, go ahead in as many RTV crops as we can. At the moment, I believe that that is feasible in cassava, in potato with the constraint mentioned, in sweet potato as well. But in addition to what you mentioned, it is our plan to move as quickly as we can into spar testing so that we make progress not only on the genotypic end, but also on the phenotypic end as well. Over and thank you.
Thank you, Hugo. Maybe I can just add one thing. So when we plant, for example, cassava seed, we get a single stem. So that is the stem that we will clone to go to stage 1 testing. And at most, you can get 5 stakes or up to 8 stakes, and that's just sufficient for a 1 row plot. Now, then we can play around with different strategies to do this fast testing. Like, should we plant smaller plots and split this across locations, or split families across locations and then use the checks for interconnectivity? It becomes sort of like an engineering problem that needs to be well designed and so that we can maximize the coverage of the TPE as early as possible. And this is something that we're actively working on, Gary. Thank you. Anybody else? I can see a lot of friends here. If you don't speak, I'll call on you. Bodo, we didn't hear you today.
Thank you for this nice invitation to say a word. Good afternoon, everybody. Yeah, it's quite exciting what has been going on there, and quite exciting how these different ideas that were shaped already several years ago by now are making progress. So congratulations. Go ahead. Just maybe one observation. Overall, I think we are the 1CG, or let's say the CGIAR. The CGIAR has been famous for its germplasm and for its outreach to national programs. And I think the best outreach would be to create advanced germplasm that is kind of pre-bred, that can be used and taken up with pleasure by the nationals. And therefore, I think that the phenotyping deserved highest priority. So to continue increasing the quality of the phenotyping. Yeah, and we've also seen that in the past, the CG may have extended a little bit into, let's say, development of disease resistance and abiotic stress response, and here and there quality characteristics might have been overlooked or set back a little bit. So I hope that this can now also be tackled much more to fix this problem. Yeah, you know, in the West, for example, where I come from, the potato must be smooth. You do not see an eye where it is not cooked at all. It's smooth, and once you boil it and you peel it, the peel comes off in one sheet. So I mean, these are quality characteristics that might need to be kept in mind. The same with cassavas. Cassava has a tapered root. I've seen cassavas in particular in Latin America, and we have our colleagues from Latin America here that have, let's say, roots that have more a round tip instead of a tapered shape. So maybe the West African breeders can learn something from there, or maybe by sharing the appropriate germplasm, this trait could be introgressed here into the material that we have here. These are just a few observations. Thank you.
Thank you. Thank you. Yeah.
Isma, this is Maria. Good
Yes.
afternoon. Yeah, thank you very much for all the presentation. I think they were all very, very good. And I'm in agreement with all the comments made. I think there is a lot of investment on infrastructure, which is quite good. Also on training, which is very good. I'm very happy to see work, a lot of this work that the Cassava, Next Gen Cassava is doing on flowering, and that is very good work that other crops may also take advantage of all those technology that you're doing, because we could have similar problems within the RTB crop. But I feel like we need to also to do some more work on the quality of those crops. And I hope also that eventually for the Southern Africa and the crops in Southern Africa could also be done similar type of investment. Thank you.
Thank you, Maria. Yeah, as you have seen, maybe a quarter or close to a half of the investment is just building the infrastructure for quality evaluation. And this is something that we're taking very seriously since the last few years. Of course, now that we know what is product profiles and market segments, and not breeding for a trait, but breeding for a product. So that's why we have bumped this as a priority. I can see Claire is in the house. He unmuted briefly. Please, Claire, can you jump in if you want to make a comment?
Hi, Ismail. I've enjoyed the presentations. I wanted to follow up on your comments about the possibility to split the stakes from that single F1 plant and send to different locations, which Gary was also commenting about the possibility. And at CIAT, we in fact did that since the mid-1980s, because normally in that phase 1 selection, you're eliminating 90% of the variability in a single site. So it makes a lot of sense, even if you've only got a plot of 1 or 2 plants, to send that out to 2 or 3 locations where you have significant variation in your key selection traits, diseases or soil types or whatever. And that was very successful for us at CIAT, and continued, I think still continues. So it's complex to manage. A lot of packs of stakes, thousands and thousands of packs of stakes going out to different locations, but done carefully can be done and very successfully. So I encourage programs to do that.
Yeah, good to hear from you, and we have taken note of that. Elizabeth, I saw you're unmuted briefly.
Yeah, thank you, Dr. Rabi and all the presenters. Yes, a very good presentation and a lot of good insights and intervention. I was also thinking that this project, the Crop to End Hunger, had a strong gender component. So I'm just admonishing everyone to, like we keep an eye on that. Do no harm, and then make sure at the end of it, that would be realized. Then also with KEPHIS, I was wondering whether there's any security component thought of, because all other programs are on, like secured premises. So I was just here looking at what she presented, Morak, but I'm just wondering, maybe it was there, but I didn't notice. So something to do with security. So that is important to consider. But I think the insights we have received are good, and we would also, look at it and see where I think we can consider, because I know Henan would have from silliness, we will have about 6 cartons, and then spread it like Dr. Hesse just said, in locations, and that's how he did his, screening. We can really ruminate over it and think about it deeper. It may come with some complexity, but maybe we would learn a lot from that. Thank you, and thanks for this presentation. And so also we see that genomic selection is getting to our NARS partners everywhere, the way we are going. And so it will become something to be routine that they can use. Thank you for the presentations. Thanks.
Thank you, Elizabeth. Yeah, do we have any more comments, suggestions, questions while you're preparing to raise your hands? I just want to thank the CtEH funders for giving us this opportunity and the resources, because traditionally, when we write grants for projects, for example, for breeding projects, it was very difficult to justify upgrade of like facilities like buildings or major renovations, because I thought maybe they assumed you as an institution, you should already have those. But some of our institutions are 50 years going on to 60-year-old, and we were really struggling. So this initiative, I hope it's not a one-off, and we're gonna, if we show some good output in terms of delivering better genetics and shortening the breeding cycle and increasing our efficiency, that we can get more investments in terms of infrastructure and facilities and equipment investment. So you know yourself, those of us, those who are in the room who have been helping us with this. Also, Gustavo and basically the EIB team have been very, very helpful, brainstorming, discussing on these engineering issues, making recommendations, and also even linking us up with designers who can give us advice on these workflows and so on. So we're very grateful for that.
Yeah, I think it would not hurt for me to also add my voice to say a big thank you. Now, we realize with KEPHIS that occasional upgrades are important. So like what we are doing in Ibadan, maybe after a while, we'll to have more, search grants to make sure, things are up to they are very good and performing efficiently. So we are so grateful, and we really appreciate the support. We don't take it lightly. Thank you so much, Dr. Rabi.
So if there are no more comments, questions, I think it's clear. There's no harm in finishing earlier than on the schedule.
Hugo has a comment.
Yeah. Ismail, I just have another comment because when you mentioned that we need designers just to get the buildings right and the flows, correct?
Mm-hmm.
So I think that we don't need to reinvent the wheel. The most effective way to get that is just reaching out to companies. I mean, Corteva, Bayer, KWS, they are building all the time. They have the expertise or the right consultants, because we might come up with the best engineering company in Kenya or in Nigeria, and they are curious about what we do. So I think that the most effective way would be just to reach out to our friends in companies and asking for their advice. I'm sure that they will help us, because again, they do this on a regular basis. So they design buildings and create a flow of work, not just from a purely engineering point of view. They do it from a plan building standpoint, and that makes the difference. Over. Thank you.
Thank you. Point well taken. Yes. Cool.
Ismail, can I just ask you quickly, as a follow-up to your point about showing genetic gain, the need to show that we're finding that our leadership is really interested in seeing the impact of breeding investments now in farmers' fields, and we do need to show we need to work on ways to show the rate of genetic gain we're generating on farm in sub-Saharan Africa. Can I just ask quickly, what is the status of on-farm testing? Do we have extensive on-farm testing in RTB crops that we can use to estimate gain on farm? What do we need to do in that area?
Thank you, Gary. Maybe I will give opportunity to the other TB colleagues to jump in if anybody wants to answer that.
Yeah, yes. Let me start, but I'm sure that others will want to contribute as well. So, Gary, you raise a critical point. At the end of the day, the only real genetic gain is the one delivered on farmer fields and not on all research farms. And recently, we have greatly expanded our ability to do a more comprehensive sampling of the TPE, if you will, through tricot. Okay, and Chidoji, Ismail, and others can tell you that the number of tricot locations, there are so many more than as before. And in sweet potato, we have done exactly the same. Now, the question is, to what extent is tricot data acceptable? Because it's still a relatively new approach, but I believe that that's the way forward. Because otherwise, we will just for logistical and budgetary reasons, we just won't be able to sample our TPE at the level of granularity that, for instance, a breeder would do in the US. But to tricot, we have gone from just a very few number of locations, which frankly, they are hopeless, into these numbers, still far from perfect. But I would say that, I mean, keeping on improving tricot information. Again, I am a big fan about exploiting pedigrees and genomic information. If somehow could help us to somehow further expand what we get out of tricot, I believe that then we will get into a level of effort that will give us enough confidence to give you an answer. But I'll stop there. Thank you.
Thank you, Hugo. Anybody else want to jump in before I also give my take?
Well, I can just make a general comment, Ismail. I think it's an ongoing discussion, Gary, within the RTP breeding team. We have had some experience, I guess, in many of the crops. I could talk about potato and sweet potato. We are looking into the information we have. I think a lot of what we have done in trichome, for instance, up to now, we had in the mind of the idea of promoting the advanced clones, not really thinking about how we could use this information to measure genetic gain. There is a big effort now. I was talking to Jacob and also to Kawe, for instance. So they are working to find out what would be the statistics that we could use, using this rank that we are collecting, so that we can leverage from it and start to think about or to measure what is the impact that we are having on farmers' field, right? From the most advanced materials that we are having. And we are also discussing how we can, of course, take to another level this kind of on-farm trials on RTB. I think this was a topic of our discussion in our last meeting, or our meeting in Uganda. And I think it's about time how we can leverage from the experience we have across the RTB and see how each crop will capitalize and deliver the genetic gains on farmer field. Yeah.
Mm-hmm. Yeah, I mean, I couldn't agree more with what has been said. Chidozi, you want to speak?
Just a comment about the scale we've gone in on-farm testing for RTB crops, especially cassava. A lot has been done, so it's no longer a question of are we doing large on-farm trials. The next level is what is being implemented now, being able to take quantitative data in tricot. That was in our initial, our first generation tricot had no such, and we've learned our lessons. That is important to take it. So that is now a given and makes it easier. So on-farm is prime time, I can say that. Thank you.
Maybe I can just throw back a question at Gary from try to just clarify this from my mind. So when we did first, typically on-farm trials, we used to run them when we have materials that we want to release. So we don't run on-farm trials every year. But with the, trying to make sure that our breeding testing TPE is breeding sites are covering the TPE, we are putting more and more of our late stage testing, distributing them more, and even trying to plant them under some farmer fields, those who are capable of managing those trials. And our job is just to deliver the stakes, lay out the experiment, and then they manage and we collect that data. So that is part of like the last stage of breeding testing before we go for like the true on-farm. Now with the tricot, we did the tricot because we wanted to know how farmers rank different genotypes, and from there decipher what are the traits, the main drivers that are associated with this ranking, so that we can refine our breeding targets. Right now we're using tricot in a different sense as a surrogate for on-farm testing. So getting the ranking directly from the farmers, but also delivering that data to the variety release committee as part of the on-farm testing. But I think we need a little bit more discussion on how we allocate these resources for this on-farm testing vis-a-vis our last stage testing, because it's a big investment. Usually our last stage testing with partner networks, it's easy because the partners can manage and collect the data. If we're doing on-farm testing, that means our technicians have to travel, and travel is a big budget eater, so to speak. And so it requires a lot of resources. But I think we're working with Docas and others, and we'll try to refine how we can use the on-farm as part of our testing to also include data for genetic gain estimation. I don't know if I'm making sense there.
Sure, it is expensive. You need on-farm testing or testing under farmer management when you expect the correlation between performance, the genetic correlation between performance on farm and on the research station not to be high. And in many contexts in sub-Saharan Africa, that's a reasonable expectation because farmers apply less fertilizer, they often use lower plant densities, they intercrop, their weed control is different than on the research station. So, you know, we often find, we're certainly finding in maize, that the genetic correlation between performance on the research, in research station trials and under farmer management is not that high. So, and the use of designs like Tricot, which is really using a farm as an incomplete block, and there's a long history of doing that, tracing back to the mother-baby trials in maize. Using that to allow you to move that late-stage testing on farm. So whenever you suspect that correlation may be low, and it's something we should be testing, we should be doing experiments to measure the correlation between performance on the research station and on farm. You know, we need that type of trial. The Tricot model promoted by Jakob and his team really did focus heavily on ranking and on farmer reaction to the material. We've from the Gates Foundation side, we know that is valuable information, but you know, if you're going to rank and rate a cassava trial, you're going to have to dig the roots up anyways. So you might as well put them on a scale. You know, we really need to get that quantitative data to understand the progress we're making in these programs. I agree with you, Ismail, that it's very expensive and time-consuming to send our technicians out to collect those data. So that's why the successful networks doing this, they don't use CGIAR staff to go out and do the on-farm trials. They make they do it with on the basis of partnerships with the extension system and with NGOs that are doing agricultural development work at the village level. And the CG and national program role is to coordinate, to train, to manage data, and to make the final recommendations. Just a thought on using the late stage on-farm trials to estimate genetic gain. That's not that difficult. That's a matter of putting the right checks into the trial. You need to put a few older but still used checks into the trial that allow you to do the regression of yield or other quantitative traits on year of release of the variety. So it is expensive, but it's an area that we do need to identify the resources to be able to invest in that type of testing if we have evidence to indicate that the correlation between performance on the station and on farm is not that high. So and that's another area where we need to collect that evidence.
A lot of good discussion there, and I'm sure we actively, in fact, in Nigeria, we currently have an Tricot trial as you know, working as a release on-farm trial. And we have different kinds of check, including old improved, new improved, recently released varieties, benchmarks, and so on. So we'll see what kind of data we get out of that. So we have basically 3 minutes. We have to close on top of the hour. I think I'm seeing Bodo has his hand up. Bodo, go ahead and.
Okay. Just to add up on what Gary said, I fully agree. It is expensive, but it is important to get this data. And, you know, thinking about solutions to this problem. What about, I mean, it's different from this project we are talking about here, CtEH. It is something that goes into the new structure that we are trying to develop, including transform the work package in accelerated breeding, where we are reaching out much more and much more in a much more systematic way than before to our national and NGO and SME partners in the widest sense. Would that be a task for that team, for example, to help the breeders speeding up collecting back good data on the performance of those items that have been given out for testing, or also going into details of trialing on farm somewhere? Maybe this is something that we need to discuss in the near future to set it up. And I think this points to this other part of our responsibility as the CG, that is training and education of the nationals, because we are here, we are the consultants in this sense. So we are here to enable others to eventually take this job. And this is something that is now easier with this additional equipment. And maybe that is why this goal comes a little closer. Thank you.
Thank you, Bodo. I think we will take that into consideration, and of course, as part of Transform, we can discuss about distributing and restructuring this kind of activities. So I think we're almost there. We have one more minute, but for me, I'm good. Thank you very much, everyone, for making time to join. We have a full house, almost 70 people joined this webinar. Thank again for all the presenters for putting together excellent updates. And we hope in the very near future, we will be able to actually see the completion of all this infrastructure development and the training and how this will transform our breeding. Yeah, so thank you very much and adios.